Awesome. Well, thank you all for coming. Glad you decided to come learn about brain networks instead of on such a nice day. I hope I'll be able to blow away the nice day with all of the fascinating brain network stuff. So there are a couple things that I'm going to talk about today. So the first is just to talk about networks in general. If you already know something about networks, you can probably go to sleep during this because it'll be a lot of review for you. Then also about how we analyze networks, and this is networks in general, not just brain networks, so you might want to sleep through that too. But understanding networks in the brain is probably going to be a bit more interesting. And then I'm going to talk about the stuff that I'm doing with regards to network analysis and how we're using this new technique that I've developed to analyze brain networks in a way that we haven't ever been able to do before. So this is a network, and this is kind of a cool network. For all the musicians, this is actually Mahler Symphony No. 2, Movement 1. This is another technique for making a network that I developed that has nothing to do with neuroscience, but it's kind of cool. So you see, if you know sort of a bit about orchestration, you'll see you've got all of the sort of low brass over there. You've got percussion over there. You've got your harp over here. You've got your woodwinds over here, your strings right here. And they're all grouped. I wonder why that is. They're all grouped with very similar symbols. These all have the octagons, and these have the squares. These have the triangles. What's going on there? Well, I'll get to that in a bit. But this will be sort of an example that I'll come back to of the power of what networks have outside of neuroscience to convince you that they also have power within neuroscience. And here's another example. You've got the bass instruments up here. You've got your horns and trumpets, and then you've got your woodwinds. And they're all nicely grouped together with similar symbols. What's going on there? We'll find out soon enough after we learn what else networks can do. So does anyone recognize this? What do you think this is? It's the UK, but what is the network?
Highways?
Not highways.
Trains. Electricity.
Yep. It's the power grid. Uh-oh. What about this one? Some of you might recognize this one. If you could read the names. I'll read off a couple of them. DVA, SIBVL, SMDDR, AINL.
Docs. So there's a computer today.
It's C. elegans. It's sort of the model organism for all of biology. What about this one? Does anyone know what this one is? Wow, you got it. So what's cool about networks is all of these different things, whether it's a piece of orchestral music, whether it's the power grid, whether it's a brain, whatever it is, we can analyze them using all of the same tools. And lots of the same tools will have different meanings. It also allows us to take things that might not be directly related and relate them to each other. And to use large amounts of information in an efficient manner. It's also something that if you talk about this to people, they'll understand it. It's very easy to grasp what the concept of a network is, versus if you use sort of very advanced statistical techniques. You can't go up to someone on the street and start talking about your ANCOVA or something. They'll have no idea what you're talking about. But it's very easy to understand what a network is. And you can also analyze these at different scales. And so, for instance, I was showing you the power grid before. But what if we zoom into a city? Then the power grid looks very different than it does at the whole country level. And so we can look at different scales as well, and we can have subnetworks within other networks. And so there are some different terms that we use when we're talking about networks. And I'll just introduce a couple of them to you so that I can talk more easily about some of this stuff. And so nodes are sort of the main things that you care about, and then they're connected by edges. I don't know who calls nodes vertices. I've never heard that in neuroscience, at least. Links, I know instead of edges, they sometimes use that in like economic networks and stuff. I haven't heard neuroscience. But so nodes are connected by edges, and that forms your network. Oops. And you can have either directed or undirected. So in a directed network, you have one node goes to another node, and this makes sense in things like, say, like traffic networks would make a lot of sense because you have one-way roads. Not all roads are two-way roads, and so you would have a directed network. Versus an undirected network, you would have for something like, say, a social network, where if you're friends with someone, then chances are they're going to be friends with you. So it's undirected. For brains, most of the time we use undirected just because that's the type of data we can get. It's really, really hard to get directed in networks. It would take, if you want reliable directed networks, it would take, hundreds of years worth of brain scan data, which isn't really going to work if we want the person to be alive. You can also talk about weighted or unweighted graphs. We usually use unweighted we can use either one. I will talk about unweighted mostly because it's a lot faster to compute this stuff on your computer, and you don't have to wait as long. Weighted graphs, they allow you to have some measure of something. So for instance, in the social network, you can say, well, how close of a friendship do you have with this person? And then you can assign weights to the network and say, oh, I'm a very good friend with them. It's a very big number. Or I'm not a very good friend with them. It's a small number. And I'm going to work mostly with unweighted. And they're also lots of the times easier to explain. And so the way that we talk about networks is through this thing called an adjacency matrix. And so for an adjacency matrix, if you give each node a number, it corresponds to a row and a column in the adjacency matrix. And so if you're connecting nodes 1 and 3, node 1 and node 3, it'll have a 1. But like 2 and 3 aren't connected. So you go 2 and 3, it's a 0. And this is going to be a symmetric matrix if you have an undirected graph. And so does all of that make sense? Good to keep going? Okay, so then you have a lot of different features you can look for in networks. And so one of the classic ones is hubs. Airport hubs, that's actually a real term, which is kind of cool. Hubs are anything that has a lot of connections going in or out of it. And so it's very well illustrated with airplane networks. Modules are another important thing, and I'm going to be talking a lot more about modules. So modules are, when you have a network, lots of times you get different nodes that will group together because there will be a lot of edges connecting them within the module, but not many going out. And so you see here you have like a lot of these clusters. So those are contained within a module. And you can have algorithms that will divide a network up into different modules. And you can also have different levels of modules to make things even more complicated. So, for instance, I like this middle one, the social network of a primary school. So you ask your 6-year-old, who are your best friends? And they tell you, and they're linked there. And so then you can make groups of students from that. But what scale do you make them at? Do you say, oh, I want to look for two groups of students? Maybe you want to look for 5. Maybe you want to look for, 10, 15 or something? And so depending on how closely you look and how big you want your modules to be, you can either get very large modules, very small modules, and that's sort of at the discretion of the person who's doing the analysis. And it can complicate things, and I'll get later into ways that you can avoid that problem of trying to find different scales for your modularity. And so I'm going to pause again. Everything good? Because of something, yeah?
What's the difference between a hub and a node?
So a hub is a node, but it's a node that has a lot of edges coming into it. And so if a lot of things are connected to a node, then that node will be considered a hub. Versus one that maybe only has 2 connections, that won't be a hub. But one that has 40 connections will be. And so like in the airplane network that I was showing, certain cities are hubs. So like London is a big hub for a lot of airline companies because a lot of things fly into London, Heathrow, and a lot of things fly out. Versus something like Luton isn't really a hub. It's not used to connect a lot of different things. So that's sort of the crash course on networks for the amount that we need to keep going forward. So now we get to talk about brains, completely switch gears, and how you can have networks in brains. And the really cool thing about brains, like I was talking before with different levels of analysis, like how you can analyze the power grid of a city, you can analyze the power grid of a country. With brains, you can also have a lot of different scales of analysis. But you can have a lot more than you can have with most other systems. So sort of the lowest level is gene transcription. And you can, well, technically you can make, people have made DNA networks too, but we'll ignore that for right now. And gene transcription networks are the simplest one that people commonly use. And where you have networks where the different nodes are different proteins that interact. Then you can have protein-protein interaction networks. So different proteins will be the nodes, and the edges will be proteins that bind to each other. You can have neuronal networks. So within the brain, you have individual neurons, and you can look at a really, really small section of brain and say which neurons are connected to which neurons. There was a really cool paper that just came out in Nature 2 weeks ago, I think it was, that looked at neuronal networks. They built this huge neuronal network and integrated different types of data. Really interesting. I can give you the reference if you're interested. But neuronal networks are next level. You can also have microcircuit levels. So this is where you look at small areas. So maybe you'll look at the visual cortex, or maybe you'll look at the motor cortex, or other individual little pieces of the brain. Brownie points if anyone knows what this is. It's a rat hippocampus. So it's a hippocampus and a rat. And so you can make a network of a rat's hippocampus. You can also have networks where you have a small number of regions interacting. And I was talking before about how hard it is to make brain networks that are directed graphs. When you only choose a couple different regions, you can make directed graphs, and there are some cool tricks to doing this. Dynamic causal modeling is the main one. And so you can make graphs of, if you just have a small number of regions in your brain. But the things that I mostly work with are full brain graphs, which is where you take the entire brain and turn the whole thing into a graph. And you play around with it and see what you can do with it. And you look for modules. And so there are also different ways of making a whole brain graph. So you can either use correlations in activity. You can use different components of the structure. There are lots of different techniques within MRI that you can use for getting at different aspects of the structure. So you can image the myelin. The myelin is the thing that wraps around your neurons to make sure that they can send electrical impulses to each other. So it helps them communicate. And so you can look at that, which will show you where the major connections are within the brain. You can also look at there are a lot of other ones that don't necessarily have as firm of interpretations yet that you can look at. You can even look at whole brain gene networks. Those are pretty cool, where you look at correlations in gene expression data across different aspects of the brain, different parts of the brain. And so for most of the time, I'm working with the correlations in activity networks based on that. And so what you do is you essentially give someone a brain scan and tell them, lie in the brain scanner for the MRI machine, lie in here for 15 minutes or 10 minutes. And just let your mind wander. Don't really think about anything. Don't fall asleep, but either keep your eyes open, keep them closed, and just sit there and just think about whatever. And we call that resting state, fMRI. And then over time, we look at the changes in blood flow in their brain in different regions. And this is supposed to be brain activity, is what it's generally referred to as. The bold signal is the technical name for it. And so what we do from here is we condense down the time series that we get from here. So for each of different regions in the brain that we select ahead of time, we have some type of time series that describes how the brain is activated over time, this bold signal, how it changes over time. And so each of them has its own time course that goes along. And from that, then we get one for each brain region. So let's say we have 500 different brain regions. So we have 500 different of these time series, and then we take the correlation between them. And so then any two given regions, we find the correlation. And so when you do this for all possible pairs, you get a correlation matrix. And a correlation matrix, so where each different region corresponds to a different row. And then from that, you apply some type of threshold to it. And from that, you get another matrix of zeros and ones. And where have we seen a matrix of zeros and ones before? Yep, our adjacency matrix. So from this, since we can go from a graph to an adjacency matrix, we go from the adjacency matrix to the graph. And now we have a brain graph. Ta-da! Pops out of after this really long analysis. And some steps that I skipped because they're denoising and motion correction and all of that jazz. But the general idea is you look at the correlation in brain activity among different brain regions, and the regions that are highly correlated, and you call each brain region a node, and you connect the highly correlated nodes via an edge. And so an edge here describes a strong correlation between two brain regions. And so that's all great. We got a brain graph. What do we do now? Does anyone know what kind of brain this is? I'm giving you a hint. It is a brain graph.
Rat.
Really close.
Mouse.
Yeah. It's a mouse. And so I'm going to stop one more time, make sure everything makes sense, because if it doesn't, you might be confused. Yeah?
Do you have neurons in your brain matrix?
Like
Or
individual.
Or you just analyze the region
We just look at the region.
and not neurons?
Yeah, because if we were to get neurons from the entire brain, it would be, we wouldn't have any way of acquiring that data because it's too precise. It's too hard to get. This is kind of the best that we can do with what we have, and within a reasonable budget as well.
Okay, so it's based on the blood flow in each region in the brain.
Yes. And so we do that, and we get these graphs. And from there, remember modules? So we get to play with modules again now. So these are what some modules look like in the brain. It's sort of debatable depending on what scale you look at. Like I was talking about before, how you can adjust the scale and look for big modules or small modules. You get slightly different ones. But this is pretty typical. This is back here, the occipital lobe, the visual cortex, and that pops out really easily. You have sort of your the motor region, so that's responsible for taking in sensory information and like allowing you to move. That's sort of when you see in textbooks the little homunculus of the person with the really big tongue and the really big hands. That's all right up here. And then you've got the sort of frontal parietal region. So the frontal is up here, and that's sort of cognition and thinking and memory and all of the type of stuff that most people care about when they think about brains. And yeah, so those are the modules we end up getting. But it's not perfect, but there are a lot of really nice things that modules allow us to do. So most critically, and what the primary purpose of them is they allow us to see very clearly at a glance which regions of the brain are sort of associated with each other and which ones sort of work together, which ones are oftentimes involved in similar tasks, activated at similar times when people are just sitting there. It also gives us a way of sort of organizing the information that we have about what different things in the brain do. It has a lot of similarities to dimensionality reduction techniques like principal components analysis. It's most modularity algorithms are based on. You can also see that the modules that you get actually do have significance within the brain. They correspond roughly to anatomical regions that matter, and they also correspond to things that are biologically relevant. They correspond, there's one network that we know is responsible for resting state. Sorry, I shouldn't use the term network. There's one pattern of activation we know is responsible for resting state, or when people are just lying there. That's constantly active then, and it's never active at other times, and we see that pop out in these modules. And the other cool thing is that across different types of networks, so I showed you that big list of different ways that you can make networks out of brains. Most of those different techniques have similar modules, which is kind of interesting. And it also helps you find, it allows you to do other types of analyses. So there are some types of analysis where you say, okay, I found the modules. Now let's find when modules change from one module, or when nodes change from one module to another. Is that clinically relevant? Is that important? When does it happen? Does it happen in certain diseases? Does it happen as people age? And so it allows you to ask a lot of other questions. But of course, modules also aren't perfect. We do have problems with them. One of the big ones is you never know what algorithm to choose, because no matter what algorithm you choose, it'll give you different modules, which is a problem. And possibly, according to some, the bigger problem is that if you use the same algorithm, it'll give you different modules because they're stochastic. And so you never really know what to do. Do you use the first run, the second run, the third run? There's also no real modules that you can compare them to. So you can't say, oh, well, I know that this algorithm is better because these are the modules that we hope to get, and we got them. It doesn't really work because there's no ground truth that you're comparing it to. And there's also something called the resolution limit. So I talked about how you can look for big modules, you can look for small modules. The little bit of a lie there is that if you get too small, you can't look for modules anymore. No matter how you tune these, they can't find small modules. And that's a problem. You also can get, like I was talking about before, when you get at different levels, you have to tune parameters. And so the best way around this is this technique that they came up with, where what you have to do is you have to take these algorithms and you have to run them across the entire space of parameters, or everything that you can ever get. And once you get that, you get a ton of different modules. And so they say the technique goes where you don't actually analyze modules, you analyze probability distributions over modules. And that gets really confusing. Because the point of modules is that you're supposed to be able to think about things more easily. But that just makes it even harder to think about things. And the modules also have to be separate. Most decent algorithms don't allow you to have overlapping nodes. And on a related note to that, some nodes don't really fit into modules very well. And this is what I'm interested in. So imagine you have sort of a perfect case scenario where you have two different networks. So this is called the Karate Club network, and this is probably the most famous. Oh, sorry, yeah.
So regarding the problem with collecting directional data, so basically what you're saying, because of how you collect the data, like computationally, it is therefore impossible to see the time difference between the signal being sent and the signal being received. Because what you're suggesting is that if what you're saying is the brain activation, and you seek for a correlation, then presumably if area 1 is communicating with area 2, then the correlation is time shifted. And therefore, is that difficult on a computational level to...
So in theory, that's a problem. In practice, it's not, because the time constant is so small on this.
Exactly.
Or sorry, so large, not so small. And so you have, so these, we usually get them at about like 0.25 hertz. So when you're at 0.25 hertz and an action potential takes on the order of milliseconds to propagate, it doesn't really matter. Yeah. And even if you do sample faster, blood flow, it takes longer for your veins to dilate within your brain and let more blood through and that type of stuff. So
this is because you try to build a model only based on blood pressure information. You don't you mix this with other information like electrical impulses?
So we can't. And so all fMRI technology that exists in the world is based on this blood flow, because that's the only signal we can reliably get. There are other things like EEG, which is where you put the funny hat on. That is, well, so number one, it can only get stuff on the surface, versus MRI can get stuff deep within. And more importantly, it has very low spatial resolution. So EEG, essentially, you have 128 electrodes or 256 electrodes on your head. So your head's covered. But really, the only information that you're getting is you're getting like activation here, and maybe activation up here. So all of that really has a much smaller, has very few degrees of freedom. You're collecting a lot of data, but the information you're getting from that data is very small in EEG. Versus in fMRI, we have a lot better spatial resolution, and so we can distinguish these different regions. So when we have modules, some nodes don't really fit into the modules very well. And so this is the Karate Club network. And it's really famous because there were these researchers that were studying a karate club. And I don't know, maybe one of you know this. A karate club is just like a gym, and people who are in the gym, I don't quite know. But they were studying this karate club, and they said to everyone in the karate club, okay, tell me who are the set of people that you're closest with, that you're best friends with, that you know the best, that you practice with the most, that type of thing. And so they figure out, and they're analyzing their data. And while they're analyzing their data, something happens. The two leaders of the karate club get in a fight with each other. And in the end, the karate club splits into two clubs. And so the researchers are like, yes, we have awesome data. And so they started analyzing it. And they found that just by looking at the data, just by looking at this network of the data, you can predict using modularity algorithms who ends up joining which club when they split. And they can predict that from the data that they got from before they split with almost perfect accuracy. And so these are the two leaders of the clubs. And so the clubs split, and you can kind of see these groups that form around here. And those, so that you see the squares and the circles. And the squares are one module, and the circles are the other. And so here, network modularity predicts almost perfectly who joined which club when it split. But there are some edge cases. And this is why it's so important that we have to know when someone should be in a module and when they shouldn't. So here you'll see this yellow one. So it turns out that yellow guy, the module sorted him into the square one, but it didn't do a very good job because it turns out that he only has two really good friends. One of his good friends is the leader of this club, and his other really good friend is the leader of this club. So which one does he split into? I mean, it would make logical sense for him to go into either one, and there's no possible way that the data can tell you which one he goes into. And so the algorithm kind of just has to choose one. No matter what algorithm you have, there's no principled way for it to make that decision. And so it'll just choose one. But how do we know which ones are the nodes like that could have gone either way? And how do we know which ones are the group leaders that defined the module, or these people that were only friends with the group leader? So we need some type of metric for doing that. And so I created a metric called versatility, and that will tell us exactly that. It'll tell us which people are likely to join, or which people are sort of on the fence, or which nodes are on the fence between modules, which ones don't quite belong to any of the modules that are in the network and just kind of float in between. And so calculating it is fairly straightforward. So you've got your graph that you start with, and this is, it can either be the karate graph or a brain graph, and like for instance, you have this graph. And so you can see how this is probably a module, and this is probably a module. But what about this guy? It's connected over here to 2 and over here to 2. And so what you do is you just run the algorithm a bunch of times and see what happens. And so you run it a bunch of times, and as we expect, the person in the middle sometimes ends up over here and sometimes ends up over here. And so you make a new matrix from that. And for each pairwise combination of nodes, you say, what is the probability that they're in the same module? It doesn't matter which module they're in, just the probability they're in the same module. And then you plot that matrix, and it looks something like that for this. And so you can see this gray row in the middle. The only one that it's always in the same module with is itself. And with all of the other ones, it has about a 50% chance of being in either one. And so then what we do from that is we say, okay, we only care right now about the ones that can be in either one. We don't care about the ones that are always together or always apart. And so we apply a transformation on that, a nonlinear transformation, which in essence is just like the first hump of a sine wave. If you like information theory, you can use entropy, Shannon entropy. It doesn't really matter what you use. You get pretty similar results no matter what. And you get another matrix from that. And this new matrix will tell you which individual edges are not always there, are sometimes there, are sometimes, which individual pairs are sometimes there and sometimes not there. And so from that, you just sum over the rows or columns and normalize it, and you end up getting the versatility metric for each individual node. And so you get high versatility for this because it flips and flops between the two, and then low versatility for the other nodes because they're always in the same module. And so essentially the take-home point is that versatility will allow you to see how well nodes fit into modules. Sometimes they fit in really well, and sometimes they're the social butterflies that just like to go around and talk to people in all the different modules. And so it turns out versatility is also really useful for a lot of problems. I've only looked at the neuroscience ones, but there are a lot of other things that it can be useful for. So one of the first ones that I looked at was study of autism. And so if you look at a data set, you can get data sets where you have resting state brain scans from a bunch of people with autism and a bunch of people without autism that are age matched and all that jazz. And so you do a modular decomposition on them. So you say, what are the modules, essentially? And you get the modules, and it turns out there are 5 of them. And there are 5 of them in both cases. And then you can see, okay, so these are the modules over here, these are the modules over here. Which nodes are in different modules in different cases? And so this diagram shows over here you have your healthy controls over there and autism over here. And so in healthy controls, you have some in module 3 that then jump to module 4. You have some in 4 that jump to 3. And these 4 jumping to 3, that's the biggest trend that are in this particular data set. And so you can say, okay, so I see that they are, I see that over here we have these nodes that go from module 4 to module 3, but does that matter? And is it important? And most importantly, are these nodes actually closely affiliated with module 3 here? And are they closely affiliated with module 4 right here? Because if they're not closely affiliated, this means nothing. And so you can use versatility to say, well, I'm not going to believe this result unless it turns out that these are closely affiliated. So it allows you a check to make sure that you're actually using your data in the way that it should be used, and that what you're seeing isn't just an artifact of the algorithm. And it turns out that a lot of published work uses this same type of approach, and they don't check for something like versatility, because I haven't published versatility yet. And so when you actually do an analysis like this, there are lots of analyses exactly like this that report these results. But this, it has very low versatility in the healthy control case, and also very low versatility in the autism case for this particular set of nodes going to 4 from 4 to 3. Same with the ones going from 3 to 4, the ones going from 3 to 1. Almost all of these, almost all of these have very high versatility in one of the two cases, which means that they're not very tightly affiliated with their module. And so you can't really logically say this is important, this is something we should care about. Except one of them, which is really interesting because it's autism and it's the FFA, so the area that recognizes faces. And that one is reliably changing from the visual network to the attention network, which is something that you would actually expect to happen in autism. And that's the only one that's significant. Well, significant in terms of having low versatility values. It makes sense. And none of the other ones, like when you look at it, conceptually make sense like that one does. So I found that result really cool. Another result, so I was talking before about how you can't really, there's no easy, good way to choose which scale you look at when you look at your modules. And so like here, you don't know, do you analyze it at this scale where you have only 2 communities or 2 modules within the primary school network? Do you look for 5 of them? Do you look for 15 of them? How many do you look for? So what you can do is you can say, how about we look at the one that generates the lowest versatility? So in other words, the modular partition that's the most stable, the one that when you look at how tightly the nodes affiliate, they're all really tightly affiliated with the modules that they're in, which makes perfect sense. You would want to look for the modules that are the cleanest, the most tightly integrated with each other. And so what you can do is you can look for the values that have the lowest versatility. And so in the mouse brain that I wrote rat there, that's a mouse network. In the mouse network that I showed you earlier, you can see this sweet spot right here where this whole region has very low versatility. So you can say, let's look at this parameter value and look at modules that are kind of small, smaller than usual, but not all that small. Usually the modern way that people do this is they use gamma value of 1.0 because that's the default in every single piece of software. But you see that 1.0 actually has really high versatility, and it would be much better to use 2.0, 2.1. And those give better modules for mice. Another thing that's kind of interesting in the brain is that the areas that have high versatility are also the areas that are responsible for learning, cognition, thinking, all of these sort of higher order human type of things. What makes us human? So in mouse, in mice you can do genetics, which makes it easier. You can't do that very well in humans. And so in mice, you can look at a mouse brain and say, what has the highest versatility of all these regions? And then you can look for gene enrichment, it's called, which is where you look at, of the regions that have high versatility, which genes are most commonly expressed in those regions. and they're the genes that are related to the three. So you get keywords popping out of this when you look in these genetic databases. And so cognition, learning and memory, and single organism behavior are the three that are most common. So these regions that have high versatility are also responsible for high-order cognitive processes. And you can't do the same thing in humans because we don't have as great of genetic data. But you can look at myelination. So myelination is one of the last things that develops in humans. And the place that it's last to develop is in these areas that are responsible for sort of functioning. Myelin is also the thing that keeps your signal. It insulates your fibers, so it stops the electrical activity from spreading to outside portions of the brain, which intuitively you would think, hey, that's probably related to how well they keep within their module in networks. And it turns out that is the case. And so the regions that have high versatility are the regions with low amounts of myelin, which is also pretty exciting. And of course, another application is all of your fields, if you use networks in them, there's probably going to be a really cool application. This is true in, I think, all of the fields of everyone here. There's something really cool you can do. Even humanities fields have cool stuff with networks. And so let's go back to our orchestra networks, or the band network. And so depending on your knowledge of sort of orchestration and how orchestras are divided up, you might know that there's like lots of times the woodwinds work together, and the high brass and the low brass and percussion and that type of thing. And so here, what you look at, the different symbols are different modules. And this is when you make a graph based on the correlation of rhythms of instruments in the orchestra. And when you do that, you look at the modules, and the modules look like you expect them to look, where you get a module for the high woodwind instruments, you get the bass section, and then you get upper brass, and then you get the low brass. But what's really cool is that versatility also works exactly like you would expect it to work. So the low brass and tenor sax, which is oftentimes has the same function, they're kind of split. And they have very high versatility because they don't really fit in with the basses. They don't really fit in with the high brass, and they don't really fit in with the woodwinds. And so they're kind of in their own. So their module is officially affiliated with the basses, but they're not very tightly affiliated with them because they're not bass instruments. Same thing with the Mahler piece. And so this is the Mahler Symphony 2 Movement 1. You get the percussion over here, which has very low versatility. They're colored by versatility. And so you've got very low versatility over here with the percussion because they're not really associated with anything. You've got your low brass over here that also has very low versatility because it's a very compact sort of system in its own. But down here, it actually tells you something about the orchestration patterns of the composer. And so you've got the strings module over here, and you've got the woodwind module over here. And you see right here, which is the first violins, kind of go with the woodwinds, and they kind of go with the strings, which is exactly what you would see in orchestra pieces and what makes sense. And same thing with the horns. They kind of associate with the brass and kind of with the woodwinds. And traditionally in orchestration, they're considered to be the bridge between the woodwinds and the brass. So the orchestra piece works out exactly like you would expect it to. And so there are lots of other cool things that we might be able to do. We just need the data. We need money to pay a grad student to do it. And thanks to a lot of people. Ed Bullmore is my PI. My lab's the brain mapping unit. Churchill Foundation is the cash. Lucy organized the whole thing. And the MCR committee was also, I think, some cash. Probably for the cheese. Obviously the most important part of tonight. So thank you very much for listening.
I have 3 questions. 2 are stupid, one of them is a bit less stupid, hopefully. So, starting with the stupid questions. Between healthy humans, or I guess what we perceive as healthy humans, what is the variation that you get in this sort of thing? Is it considerable?
The variation within versatility?
No, the variation within the readings you get, very simply.
So the actual versatility values are the raw
The raw mostly,
fMRI.
just like, how much do our brains vary? Like on a normal group of human beings.
Like, do you want numbers, percentage of variance explained by regression?
I would be happy with
like-
a lot or not so much.
So it's somewhere in the middle. There are some things that are always consistent across people. Not necessarily when you look at the time series, but when you start to look at the graphs. There are a lot of things that are always very consistent.
Would you compare that with like higher brain functions? Or
Yeah, well, so some things are always consistent and some things are not.
right.
And the things that are not, some of it depends on individual variation. Some things depend on the parameters that you're using to scan. So the parameters of the scanner and apparently what time of day you scan people at, when they last had caffeine, their mood. Lots of really small things influence it as well. But there are a lot of trends that always pop out no matter what.
So the second stupid question, how are you making the edges between the musical instruments?
So what I do is I look at, so I take the score. So instead of taking the recorded piece of music, because that doesn't give us enough information, I look at the original score with all the instruments, and I just look at the rhythm that each instrument is playing. And then I make delta spikes in a time series, and then correlate the delta spikes.
Before we go, as a just practical consideration, how did you import that from the score into a time series? Did you have to get from the PDF to the time series? Did you actually type this out, or what did you do to?
I didn't go from a PDF. I went from, let's see, this one came from a music XML file, and this one came from a MIDI file.
Oh, okay. Yeah, that makes sense.
So MIDI is kind of, in some ways, it's perfect. Well, you would think it's perfect, but it's limited to 32 channels, which is why there are, so not in this one because of the way they coded it, but in the movement that has the most number of instruments, it's exactly 32 instruments.
And so there's just a channel for each instrument and
Yeah, well, so they had to combine instruments in here.
Okay.
And so technically some of these, because Mahler Symphony No. 2, it's a huge orchestration. It's what I think, 8 trumpets, 10 horns.
As it should be.
So they did combine some of them, which is why you only have 2 horns. But that's a limitation of MIDI.
I was just thinking through the nightmare of trying to go from a PDF score.
And the third question, how does the variability vary depending on your algorithm? Because obviously how you presented it, it seems that the probability, whether it's in a module or another, is very strongly based on what algorithm you are using.
Mm-hmm.
So how dramatically do different algorithms change your result?
That's the awesome part. And so I didn't show that data. Maybe I should have put that up there. But so the algorithms will give you completely different modules. But when you look at the variability, there's a really high correlation, like R squared of around 0.7. So there's a very good correlation between algorithms that give you completely different results, which is really interesting, and I don't fully understand why that is.
So if I've understood it correctly, this is a stochastic process. So you're running it with lots of different initial probabilistic data, and then basically generating an average of these to calculate versatility. Is that right?
Yes.
Couldn't you do the same thing over parameter space as well? Where you've got different parameters inputted into the algorithm, like different kinds of algorithms, do the same kind of thing, get an average, I guess. And, if so, would you not have to weight it as well? Because, from, algorithms I might use, like parameter spaces. Some of them are really terrible, and some of them are going to be much better. And it's kind of obvious which ones are going to be. So you would want to weight against them, I guess.
You could, in theory, weight on different, use different parameterizations and different algorithms. So there are two different questions. So in the case of algorithms, it would make the metric weaker because, of course, algorithms are going to give you different modules, and there's no way to match modules. And so this is another problem. One of the things, a technical thing that versatility overcomes is that it isn't dependent on the modular structure itself. It's only dependent on the algorithm. And so when you mix algorithms, you have to do matching of the modules, and it becomes module dependent, and it doesn't turn out well. For parameter spaces, you can, when I call things versatility up here, I'm not using versatility at a particular parameterization. I'm doing an average across a bunch of different parameterizations, because that also makes it independent of the parameter.
Yes.
And so you can get rid of a lot of variables by doing this.
So then I guess that ties into my second question. Do you want to weight that average at all, or do you just, across the parameter space?
You can, so it doesn't really matter. You can, and technically it would be more correct. Right here I'm using linear spacing of parameters, and probably some type of log spacing would be more appropriate. And you can make all of these tweaks and stuff, but I tried one case where I just randomly chose a bunch of values. And by randomly, I mean human randomly, so not with a random number generator. And so I had a big cluster somewhere of like 0.1, 0.2, 0.3, 0.4, 0.7, 0.72, 0.75, 3, 3.1 came out the same. I mean, very high correlation. So.
Can you go back to the application 2?
Application 2.
Yes, so the graph on the right, if I understand correctly, the x-axis is the scale, and the y-axis is the versatility.
Yes.
And it seems that this one isn't revealing enough information because I don't see any pattern. And what I expect is that there is an optimum, very obvious optimum scale that you can choose. But for this one, you say one is not good, you should choose 2 point something. And I think it has so much fluctuation. Is that because of other reasons other than the scale of the module?
So it's because I don't have enough. I only sampled this at 0.1 for each of these. And so this is also, so some of them turn out better than others. And this one is actually one of the worst ones. This is about almost as bad as it gets.
Okay.
The reason I used this is because I used it previously. Lots of the time what you'll get with this, and this isn't the case all the time, you'll get something that goes here, it goes up, it goes down, and it goes up again. These low values we can disregard because that's when everything is in one module, so it's not interesting. But lots of times we get these big things like that. And when you look at a lot of these, you start to see patterns like this. And so when you smooth this out, you see that it looks something like that after you stare at these for a really long amount of time in a lot of networks. And so this is kind of our sweet spot because it gives us a range of parameters anywhere from about 1.8 to maybe 2.4 that'll work approximately all equally well and work better than these. Like there's this 1.6 that works well, but if you go slightly lower or slightly higher, it's going to give you something wacko, and so you don't want that. So this is sort of our stable area where we can expect to get something reliable. But lots of times I won't go look for it now, but lots of times you'll get something that looks like either that or that, and there'll be a very, very clear optimal. It's less clear here, but that stable region would probably be considered a good place to go.
So at the moment, what I can see you've done is you've taken a tool, come up with a really nice new tool. You've applied it to lots of different data sets, and you've seen positive results. In my field, that would have saved a lot of time if people had been doing that. They didn't. Never mind. They kind of jumped more to the maths and the statistical analysis of the algorithms. Have you thought about, convergence properties? Like if you sample over a huge stochastic, a huge amount of data, would you then converge to the true solution on any graph, or what kind of, have you done anything in that respect?
So with regards to that type of thing, I think there are two things that would be of interest that I've done. So one is the case of how many iterations do you do? So, so something like this, how variable are maybe actually over here. So like you might say, like maybe this is what brought up the question is, am I just not sampling enough when you get this variation type of thing? But it's stable. So the variant or standard deviation is about 0.01 for 1,000 samples. For 100 samples, it goes up to about 0.05, and when you get less than 100, it shoots up. And so if you take 10 samples, the standard deviation is something like 0.4 or something. So not very reliable, but once you get out to doing a bunch of different samples, once you do, so my default that I sample at most times is at 1,000 samples, and that gives me a standard deviation of about 0.01. So it's not a mathematical analysis, it's more of a practical one. And the maximum, when you look at the maximum too, it's about like 0.04 is the maximum deviation when you have two separate runs of the same thing. So that's one thing you might be interested in. The other thing would be in constructing the this process. For all of this is all done from first principles, and it's done using a bunch of model networks, expected behavior in these model networks, and what type of thing would we need to give us that expected behavior. So this is in order to construct this, I did a thorough review, or what do you call it? A thorough scan through all of the different possible combinations of things that would give you something like versatility, and selected them based on a set of axioms that I wanted it to satisfy. And so it has a solid foundation in a strong, rigorous foundation as well.
I know that some people might have to shoot off, so if you want to do any more questions, let's do them in the bar over wine and cheese. So thank you very much again.
Thank you all.